In this video, we will discuss about structure of ovule of Ephedra. In the last video, we discussed about female cone of Ephedra. These are sterile brackets, and this is the fertile part which is bearing a pair of ovules. This is called as female cone, and this is female flower. This is the micropylar tube. This we have discussed in detail in the last video, and link will be shared in the description box. Now this is one ovule, this is another ovule. So there is pair of ovule in the female cone of Ephedra. Now if we see the structure of ovule, this is ovule. If we cut its longitudinal section, so what is the structure of longitudinal section of ovule of Ephedra? This we are going to discuss. We can see here, this is the stalk by which it is attached here. Stalk by which it is attached. And in the center, this part is nucellus. Now this is nucellus. This is covered by, on the inner side, by inner integument. This is the outer integument. And this inner integument is longer as compared to outer integument. This is forming a tube-like structure. This is called as micropylar tube. And this floor of micropylar tube, which receives the pollen grains, which are released from the male cone of Ephedra after pollination. And these pollen grains reach by means of wind over here. This is called as pollen chamber. This is micropylar tube, which is long, and this floor of the micropylar tube is called as pollen chamber, which receives the pollen grains. And these are two coverings around this part is nucellus, and this is covered on inner side as well as toward outer side by integuments. So there are two integuments, so this is called as bitegmic in nature. So it has unique features, presence of two integuments, that is bitegmic, and long micropylar tube. Now this part is called as micropyle. Now this is nucellus. This is covered by integuments. This is this is inner integument, and this is the outer integument, and this part is called as micropyle, where this integuments, they are not present here. This part is, this opening is called as micropyle, and opposite to this end, this is called as chalaza end. So this is chalaza, this is micropyle end, and in the nucellus develops the endosperm, or called as female gametophyte, where later on archegonia will develop or archegonium will develop. Now, how this female gametophyte develops inside the nucellus, this we are going to discuss in the next slide. Now, this is the ovule. This is an inner integument. This is the outer integument. And this part is called as nucellus. Nucellus is cellular. This is made up of cells like this. So, this is cellular. Here in the nucellus, this cell act as megaspore mother cell. This act as megaspore mother cell. Now this, all the cells of nucellus, they are diploid in genetic constitution. So megaspore mother cell will also be diploid in genetic constitution. Now this megaspore mother cell will divide. This is megaspore mother cell. This will divide into 4 nuclei. Now they will get arranged like this into 4 cells. Out of the 4, 3 will degenerate and 1 will remain functional. So we can see here these 3 megaspore, they degenerate, and this remains functional, because in the megaspore mother cell, meiosis cell division occur, and these 4 nuclei which are being produced, they are haploid. Megaspore mother cell is diploid, and these 4 nuclei, they are haploid. So this functional megaspore will have haploid nucleus, and this is nucellus. So here this functional megaspore nucleus, which is haploid, now this will divide into 2 nuclei. This will undergo mitotic cell division, mitosis. 2 nuclei will be produced. Later on, these 2 nuclei will further form by mitotic cell division 4 nuclei, then 8 nuclei. Small vacuoles, they appear later on. These vacuoles, they coalesce with each other to form a bigger vacuole. So these all nuclei, they are pushed toward the periphery here. So nuclei which are pushed toward the periphery, along them wall formation occurs. We can see here white color wall formation is being shown, that around these nuclei, formation of wall around these nuclei is done like this. Ultimately, this part, when wall formation is taking place, this part will also become cellular. So all the nuclei, wall formation will take place, and ultimately this area will also be filled, and this will become cellular in nature. So we can see here this will enlarge in size also, and all the cells are haploid in nature. This is called as female gametophyte, and this is also called as endosperm. Endosperm. Endosperm is the area which is present, or female gametophyte is area which is present over here at this part. This will be present over here. Now if I am trying to put it into here, you can understand how this is being produced. Like this is endosperm which is being produced actually inside the this nucellus part. So if I try to make here, just wait. Right. So we can paste over here. I can show you that at here you can see this will fill the nucellus part. This female gametophyte will fill the nucellus part, and this functional megaspore has been converted into this female gametophyte. So ultimately nucellus will be covered, right? So we can see here, if I enlarge it further, we can see nucellus is filled with female gametophyte, and here at this point we will see that somehow at this point here archegonia will develop. I'll use a different color. This is not visible to you. Like we can use here this color. Archegonia will develop over here, and how archegonia develop, this we'll discuss in the last video. Next video, sorry, this will be discussed in the next video. So this is how female gametophyte develops inside the ovule, and then this female gametophyte or endosperm, which is actually nourishing tissue, and this is haploid in genetic constitution. Now this part will develop the archegonia, which is toward the micropylar end, and this part will be rich in the nutrients. So this is all about development of female gametophyte inside the nucellus of ovule of Ephedra. Thank you for watching my video. If you like my video, please like, share and subscribe.